Seems it. Hi, evening everybody. Thanks ever so much for coming. It's a very big pleasure to be here. So yes, I'm a vet and I'm based at the Institute of Zoology, and that's part of the Zoological Society of London. It's the research team that's actually part of the University College London. So I'm going to talk about wildlife disease and biodiversity. Thanks. Let's see if this works. So habitat loss, land use change, overexploitation, persecution, climate change, these are all very well known threats to global biodiversity. But I think it's only in relatively recent decades that wildlife disease has been recognised as a threat in this regard. And when we're talking about disease, it's important to understand that that covers both infectious and non-infectious disease. So infectious diseases are caused by bacteria, viruses, parasites, fungi and prions. And they can be transmitted via various routes, aerosol, for example, cold or flu, fecal-oral, for example, transmission of salmonella bacteria, sexual transmission with HIV, or vector transmission, for example, mosquito bites spreading malaria. So that's infectious disease, and it contrasts with non-infectious disease. And this covers things such as pollution, predation, trauma, genetic conditions, cancer, and nutritional disorders. But it's really important to realise that there's an interface between these two. So for wild animals, if an individual animal is sick with an infectious disease, it might be weak, it may be much more likely to be predated. And if an animal has a nutritional disorder, if it's semi-starved, it might have immune suppression and therefore be much more likely to fall victim to an infectious disease. So this evening's talk is about epidemiology, and that's the study of disease in populations. And there's a few words here just that you'll have heard, but to make sure you're familiar with. Endemic disease refers to the background pattern, the expected, the known patterns of disease, so how commonly we would expect these to happen, whereas an epidemic of disease exceeds that endemic background level. And in the news, you may have heard about emerging infectious diseases, things like SARS or Ebola. And these can be caused by novel agents, things we've never seen before, but they can be caused by known agents that cause a problem in new species or in new places, or that suddenly affect increased numbers of individuals. And one of the points that I'd like to make today is that for free-living wildlife, we often know so little about the conditions that affect them that it can be difficult to identify emerging threats when they occur, and that this isn't a stable pattern. Things change really quickly. They're very dynamic systems, and so we need to monitor them constantly to see what's happening. So I wanted to introduce you to this One Health concept. And that simply means that there's an interrelationship between the health of people and of all other animal species on the planet and of our ecosystem. And we can look at that really specifically in terms of disease. So zoonotic conditions are infections of people that are ultimately contracted through contact with other animal species. But the reverse is true. Reverse zoonoses are when animals become sick with human diseases. So we can look at this very specifically, or we can be much more holistic and know that if we have a healthy ecosystem, that provides us with ecosystem services such as clean air and clean water and a stable climate and food, and that is essential for the well-being of ourselves and all of the other animals on the planet. And here's another cartoon of this One Health paradigm. And the point I'd like to make here is that I'm a vet. A lot of today will be from a vet's perspective. But there are so very many different career paths that you can take to be involved in this type of area. Medics for sure, but also ecologists, social scientists, molecular diagnosticians, microbiologists, modelers, many different arenas where you can become involved. There's a lot of work done to understand the drivers or the reasons that diseases emerge. So this paper studied zoonotic infections, again, diseases of people that originally came from animals. And in the pie chart, you can see that they were trying to categorize the reasons for the diseases emerging. And again, agricultural intensification, land use change, these sorts of things are common factors. And here's a paper published this year where they were looking at diseases that affect wildlife, and they wanted to find out if they came from livestock, our captive animals, or if they came from other wild animals, and if human activity was involved, which is very often moving animals from one place to another. And if we understand the drivers of disease emergence, then we might be able to prevent these problems happening in the future. And so that's the reason for doing this work. So I think it's pretty well accepted that disease is an important threat if you have a geographically restricted species, such as that which occurs on an island and only on an island, or if it's a critically endangered species and there are very few individuals remaining on the planet. And these are two examples of this. This is a Hawaiian honey creeper, and that's an endemic species of Hawaii, and that means it occurs there and nowhere else. And there was accidental introduction of a parasite that causes avian malaria, and also a mosquito species that can spread that parasite to Hawaii. And because these birds have evolved in isolation, they've got no natural immunity to this disease. And so there were very high rates of mortality and even extinctions of these very rare species. This is a black-footed ferret, and that's a mustelid that is native to North America, and it was thought to be extinct. And then it was rediscovered in the 1980s in Wyoming, very small number of individuals left, so a lot of conservation effort got put into safeguarding the future of this species, but two diseases are important. Threats, first of all, canine distemper virus, something that we would vaccinate our pet dogs against, and then most recently, plague caused by Yersinia pestis. And conservationists are currently developing a vaccine to protect these very small numbers of individuals against this disease. So I think that these things are relatively well known, but the point I'd like you to walk away with today is that disease can also cause declines of previously very common wildlife species across a large geographical scale within a surprisingly short time frame. And that's why disease should be considered as a threat to biodiversity. So, for non-infectious disease, I think the most important example in recent decades is that of the Indian vulture declines. So I don't know if you've heard about this, but there are 3 species of Indian vultures who have declined precipitously since the 1990s. And this has occurred because of a licensed veterinary drug called diclofenac, which was used as an anti-inflammatory drug to treat cattle. And what we didn't know at the time was that vultures scavenge the carcasses of the cattle that are left out. The cattle tissue contains residues of that drug, and that drug is very highly toxic to the kidneys of the vultures and caused them to die in large levels, over 90% loss in many populations. And now that we understood that, those drugs have been banned for animal treatment, and we have many captive breeding centres trying to recover the numbers of these once really common species, which are an important part of the ecosystem in India and Nepal. For infectious disease, there are 2 fungal pathogens or disease-causing agents which are really important to global biodiversity. So these, I think, are the 2 most important diseases. And the first is a chytrid fungus, which infects the skin of amphibians. And since the 1980s, this has caused really marked declines and even extinctions of amphibians, particularly in the Americas and in Australasia. So that's something that's very important within the global amphibian crisis that's ongoing. And the second disease is the cause of white nose syndrome. Now, as the name suggests, this is a fungus that infects the face of hibernating insectivorous bats in North America. Certain species are very susceptible. This is a little brown bat. Over 90% mortality. Millions of bats have died with this disease only first being seen as recently as 2006. And this shows the spread. It was first seen in New York State in the show cave, and it's spreading across North America very rapidly and is a cause of considerable concern. Obviously, bats are important pollinators. They are an important part of the ecosystem, and there's a lot of work trying to be done to see if we can limit the spread and the impact of this disease. And here's another extremely unusual but important example of a transmissible agent, but this time it's an infectious tumour, an infectious cancer. So Tasmanian Devils are marsupials from Australia, and they are susceptible to this very aggressive tumour that often affects their face. It's spread most often through biting. It was first seen in the mid-90s, and it's caused over 70% declines of this widely distributed species within Tasmania. And its original origin was in Tasmanian Devils. It's a nervous cancer, and because it comes from Tasmanian Devils, their immune system doesn't recognise it as a novel challenge, so they don't mount a response to it. So an incredibly unusual problem. Now conservationists are creating captive, safe populations that are healthy, and they're trying to develop, again, a vaccine to limit the spread of this disease. So if we're trying to quantify the impact of a disease, we need the vets and wildlife health people to work in partnership with biologists who monitor these populations. And there are two good examples from North America where this has been done to show that a disease has caused a very significant population decline. And the first of these is this bacterial infection that's caused house finch conjunctivitis. Now, a little bit like we'll talk about later, these are very common birds that visit backyard feeding stations in America. And you can imagine that people could spot these very visible signs of disease. And so people letting the scientists know were used to track the spread of the condition in time and space in North America. And so that wildlife disease information was pieced together with the long-term population monitoring that the biologists had conducted, and that enabled them to identify when the disease occurred and to quantify the decline in those birds that happened subsequently. And West Nile virus, viral condition of Flavivirus, a very important disease in North America. It's spread through mosquitoes, and the natural life cycle is wild bird to mosquito. But this is important because it caused encephalitis and fatality of people and of horses. So this is a disease that can affect many different species. But some birds are particularly susceptible, and that would include the American crow, very similar looking to our bird. And again, a similar picture. These are the population trends for the American crow. The orange line is when the disease arrived. The black circles are when the population drops off. So the message is that we need the vets and biologists to work together if we're to understand the significance and impacts of disease in wildlife. So I hope that that introduction has convinced you that wildlife disease is important on a conservation platform. And now I'm going to talk a wee bit more about, so this is my background. After I qualified as a vet, I first worked at one of the RSPCA's dedicated wildlife centres, and that was individual animal treatment and rehabilitation. And very worthwhile, I felt, because a lot of the problems that we see are anthropogenic or man-made, and that includes oiled seabirds, lead poisoning, often in swans, things like shot, snare injuries, fishing line entanglement. These things happen regularly, and I feel that we've got a responsibility to get involved and to try to correct them when they're adversely affecting individual wild animal welfare, and we have caused them. But it was through this work that I became really interested in looking at the impact of disease, not at the individual, but at the population level. And so since around 2000, I've worked either on outbreak investigation or national surveillance to learn about the infectious and non-infectious diseases that affect, first of all, mammals, terrestrial and marine mammals, and over the last 10 years, mostly birds, and that's what I'll talk about in a wee bit. So why do I think that wildlife disease surveillance is important? Well, as I've already touched on, wildlife can be sources of infections that can cause disease in people or livestock or companion animals. If we understand the species that have those diseases and how they're spread, then we might be able to advise on human activity to stop those problems occurring. And as I've said, disease can be important as a threat to biodiversity and conservation and wild animal welfare. And at ZSL, we're a conservation charity. So whilst the work we do informs all four of these, our focus is on the biodiversity threat. So just wanted to introduce very briefly, within ZSL, we have vets and other people involved in wildlife health across a range of departments. First of all, we have the teams of vets who are responsible for the health care of the animals at Whipsnade Wild Animal Park and in Regent's Park. And so they have to be versatile, they have to be able to treat the hundreds of species that we have in the zoo, but to do that, they use straightforward techniques, as doctors would do, as vets who are treating cats and dogs would do. So, anaesthesia, this time of a tortoise. X-ray, this time of a fruit bat. Ultrasound, that's an elephant fetus, an Asian elephant fetus, which is pretty cool. Surgery, this might be pushing surgery, but shell repair in a giant snail. So we have to be versatile. We go from invertebrate to megavertebrate. If things do die, we conduct post-mortem examinations to understand the problem as fully as possible, to try to stop these things happening in the future. We're really involved with preventative health, vaccination, health checks, worming to try and safeguard against problems occurring in the first instance. Zoos obviously have very active conservation breeding programs as part of stud books, and this sometimes requires veterinary involvement, for example with assisted reproduction, so this is artificial insemination. And research, in as much as learning about conditions that affect in partnership with other zoos, and it's not uncommon for us to work with human health specialists, particularly on the problems of great apes. So we're collaborating with cardiologists to understand about the heart disease that affect captive great apes, and there are European and North American projects on that sort of aspect. And very importantly, the experience and skills of the zoo vets are used in the field for conservation projects. And these are just two of many examples. This is a colleague doing a health check on vultures in one of those captive breeding centres that I've already mentioned because of that diclofenac issue. And then a colleague was also involved in a training workshop for Indonesian vets on how to safely anaesthetise tigers that have been caught in snares. So an involvement in human-wildlife conflict resolution, and obviously a conservation and a welfare issue. So that's what the zoo vets do. And on the Institute of Zoology side, we work a lot with native species. This is a project that's involved with captive breeding, translocation, and reintroduction of species. And when we move animals around, we don't just move the animal, we move the package of all of the microbes that it's carrying. And so it's really important that we think about avoiding introductions of novel diseases when we bring those animals and move them around. And so that's what this project's involved with, from invertebrates such as the red-barbed ant and the fen-raft spider, through birds, amphibians, the poor frog, to common dormice. And then, onto my home turf, surveillance. So, ZSL's got a track record of appealing and relying on members of the public to report signs of sick or dead wildlife to us. That has more recently been known as citizen science. For wildlife disease surveillance, it's nothing new, but it just means that it's the general public getting involved in scientific research. And from our perspective, we hope that it's beneficial to the members of the public. They get involved in science, they're encouraged to observe and engage with nature, and also we can offer advice on how people manage their back gardens for healthy wildlife. From our point of view, we are able to do surveillance on a national scale across the calendar year, which we'd never be able to do on our own. And we're involved with this type of project in a range of native species, including terrestrial mammals. This poor red squirrel is affected by this Squirrel Pox virus. This has been very largely responsible for the loss of our red squirrel across most of England and Wales, now heading into Scotland, and it was accidentally introduced with the grey squirrel. The grey squirrel from North America carries, but doesn't get sick with this virus. We run the Sedation Strandings project, so we do post-mortem examinations on whales and dolphins and porpoises that strand around the coastline. We're interested in things like fisheries bycatch and in the impact of persistent organic marine pollutants, also investigating the causes of mass strandings that can occur, for example, these common dolphins. And we're also responsible for doing post-mortems on marine turtles and basking sharks. So a range of species again. And since the early 1990s, we've been involved in amphibian and wild bird disease surveillance. We'll come back to that in a sec. So each of these schemes so far have had in common that they're opportunistic, they're ad hoc. We appeal to people, we raise profile as much as we can do, but we've got no control over the effort, how constantly people have a surveillance effort for signs of problems. And you can imagine that that can get us the risk of bias. So if there's a piece in a regional newspaper about a particular condition, that's going to raise local awareness. We'll have a spike in the reports of that condition from that area. But that's not giving us a true picture of what's going on nationally. People are much more likely to take the time and effort to get in contact of a problem that's affecting a species which is well loved, as opposed to a species which they may feel is less positively perceived. People will get in touch quite naturally about problems where they don't know the cause. However, as I've already touched on, predation victims, for example, cat attacks, is not uncommon for those animals to be sick in the first instance, and that's why the cat was able to catch it. So it's important not to assume that the animals were healthy in the first instance. And by their very nature, we hear about these things sometime after they've happened. And that can mean it's quite difficult to get good samples for examination. And in isolation, we can't talk about the impact of these conditions at the population level. So we can learn an awful lot, but the conclusions that we reach are limited, and we really need to be aware of that. And so, time for a drink. One sec. Around 10 years ago, for my PhD, I set up the Garden Bird Health Initiative. The first thing to say is it was a really collaborative project, and that's continued. So I worked with the RSPB and the British Trust for Ornithology, a network of vets, support from the garden bird food industry, working with government, and entirely reliant on the help from members of the public to do this. And our aim was to set up a national surveillance network, primarily to focus on infectious disease. We wanted to try and learn also if there were human activities at bird feeding stations which might be risk factors for disease outbreaks occurring. For example, cleaning your bird feeders, that type of thing. And so to do this, we had two different complementary but independent surveillance streams. We once again ran this ad hoc or opportunistic surveillance. Anybody can get in contact. That gives us maximum coverage, greatest chance to hear about really unusual events, but we do have this risk of bias. And so we, for the first time, set up a systematic surveillance, and that's through the British Trust for Ornithology, who have over 10,000 very dedicated members of the public who provide information every week throughout the calendar year on the wildlife that they see in their garden, whether they saw a particular species, and an estimate of group size. And we ask them to provide information each week on whether or not they saw any sick or dead wild animals of the species that they'd observed. And this gives us a structured data set, something that we can work with to identify whether the trends in time and space that we'd seen with all of those larger ad hoc reports was indeed true or representative. So through both of these routes, we get animal carcasses for post-mortem examination. We examined over 3,000 birds in the past decade or so. We do post-mortems, and we do x-rays, we grow bacteria and fungi, we look for parasites, we do molecular tests primarily for viruses, We look at tissues with histology under the microscope. And very importantly, every time we do a post-mortem, we take an archive. We keep the parasites, the bacteria, the tissues, in case we need to go back to them in the future to learn more about what's going on. So gardens are important refuges for more than just garden birds. And in the past 3 years, we've been able to raise funding to move to a website for reporting. And we also include amphibian and reptile disease surveillance and hedgehogs. Hedgehogs are in decline, an estimate a quarter in the past decade. And nobody has looked at a population scale about whether infectious or non-infectious disease might be involved in that decline. And so that's what we're doing in partnership with the BTO, the RSPB Frog Life, with funding from various arms of DEFRA and the Esme Fairbairn Foundation. So it's a web portal for people to report things that they've seen in their garden and upload photos, and that's the way to get in touch with the 2 project vets and learn about what might be going on and see if an investigation is possible. So the second shorter half of the talk is to give you 2 examples of emerging infectious diseases that have affected British garden birds in the past decade that I've been involved in investigating. One of these is parasitic and one of them is viral. And the details of these investigations aren't important. What I want to do is give you the story and explain how we went about the investigations. One, how we went about an investigation to quantify an impact, and the second, how we tried to determine whether or not we thought a population decline would occur. So, the first is Finch trichomoniasis, and I put this slide up partially as a thank you, but mostly to point out that it's really collaborative work, international collaborative work, with lots of people involved. So, there are a small number of images of disease. I don't think there's anything too bad, but I hope that they're acceptable. So, Trichomonas gallinae is the name of the parasite. It's a protozoan, which is the sort of parasite that has flagella and you can see under the microscope moving around in water. This is known as a condition which causes disease in pigeons and doves and birds of prey for hundreds of years. So this is nothing new for Britain. And it causes these lesions to develop in the back of the throat, which you can imagine make it very difficult for these birds to swallow and feed normally. So they're often seen with wet faces and drooling saliva. The parasite is killed by drying out. It's spread in saliva when birds feed one another during courtship, when adults feed baby pigeons and doves, and potentially through shared food and water sources. And birds of prey are affected when the parasite passes up the food chain, when they eat sick pigeons and doves. So that's what's been known for hundreds of years. And then, shortly after we began the project, we started to see unusual seasonal mortality of these common garden birds, the greenfinch first and foremost, and then the chaffinch. So these birds look fluffed up and miserable and lethargic. That's just a sick bird, but they hang around in people's gardens trying to take advantage of these readily available food sources. And this is a juvenile greenfinch, and you might be able to make out that it also has a messy face. It has food and saliva staining around its face. And people reported them progressing to open-mouth breathing, very moribund state, dying, being found dead in people's gardens. So the first thing that we did was very much the veterinary investigation. What is the cause of this problem? So we do the post-mortems. We can see just quickly that these yellow lesions is a very severe disease in these birds in the digestive tract. And we took samples, cultured the parasite, looked at its morphology by electron microscopy, and we used molecular diagnostics to confirm this is Trichomonas gallinae causing this disease in a new host. So that's the first step in the process. The following year, in 2006, we saw a big change. Epidemic mortality, chiefly affecting greenfinches. So, the RSPB has collected information since 2001, where members of the public get in touch and tell them about a problem, and they believe that an infectious disease is likely to be the cause. So if you look at the pattern in those first 4 years, the peak is in the winter months. And from post-mortem examinations, we know that that's probably because of salmonellosis, a different disease. But in 2005, the first year that we saw this finch trichomoniasis, that seasonality shifts, and we have just over 100 reports in the autumn of that year. If we move forward to 2006, there's a 5-fold increase, almost 1,000 reports in that 3-month period. We didn't do a press release. This wasn't because we were appealing for reports. This was driven by the scale of the outbreak itself. Post-mortem examinations over a 5-year period, the picture remains stable. It's greenfinch, it's the hardest hit, chaffinch, the second most common affected species. And when we looked at the number of dead birds in people's gardens, 40% of the people who got in touch had found more than 6 dead birds. They're weighing 22, 24 grams. They're going to be scavenged. We're only seeing the tip of the iceberg. So we had a huge increase in the number of reports and large-scale mortality at each site. So we got a lot of media attention because the British public care a great deal about the welfare of garden birds. But we wanted to look at this also to see if we were going to see a population level decline. And so this is information from the BTO's Garden Bird Watch. So here we have the months along the x-axis, and on the y-axis the reporting rate. So this is the proportion of gardens that reported seeing 1 or more greenfinches in their garden throughout the calendar year. The grey lines represent what happens each year for the decade before the disease emerged. The red line is the year of the epidemic. It's within that historical range until the autumn when the disease outbreak occurs, and then it dips, and importantly, the line remains parallel but depressed. So this is evidence that the disease outbreak in UK greenfinches within garden habitats is causing a significant decline that happened when the disease outbreak occurred. But we needed to look in a little bit more detail to do this scientifically, to compare areas, the greenfinch populations in areas of high and low disease outbreaks, before and after the outbreak. And we also needed a control, as you often do within science approaches. We chose another common garden bird. If something like climate or a food availability had been the cause of a decline, then we might expect that would also have been hit. And I reviewed all of the reports from members of the public and applied a particular rule based on the species and the time of year to say this is likely to be an incident of this disease. And then for those 2 different data sets, we looked at the distribution. Here we've got for each county the total number of households that had an incident divided by the number of human households. We're much more likely to hear about a problem happening in central London than in mid-Wales. And you can see from the shading of blue that the hot spot was in the centre and west of the country, and so we defined areas of high and low disease incidence to work with. But before we went forward, we went to that systematic data set. These tiny yellow dots are all of the households that participated and gave us information. The red dots are where the disease happened. So in that high area, 1 in 10 of the people saw sick finches. In our low area, none. So we used these for the analysis. And then we went, so it's probably the most complicated figure, so I'll walk through it. The breeding bird survey is done each summer, and it uses transect surveys to count our common British wild bird species and generate relative abundance estimates. So not the actual numbers of birds, but relative to a 1 in 1994 when that began. So we have the high disease region, the intermediate, and the low. And we have these box whisker plots. For the greenfinch, the species which we predict will be hardest hit, the chaffinch, the second most commonly affected species, and the dunnock, which is our control. So these box whisker plots represent how much those populations in those areas have varied for the 10 years before the disease outbreak. And the black circles are what happened after the first year of the disease epidemic. And so the take-home messages are that we have a population decline of 1/3 of the greenfinch and 1/5 of the chaffinch in the high disease region in 1 year, and that for the Dunnock, our control species, those black circles are within the plots. So that's compelling evidence for the population decline being due to this disease. The disease has moved around. We still see it. It's ongoing. The population declines are ongoing. Here's an update, 2006 to 10. So this is for the UK as a whole now, not a little area. The greenfinch had been increasing. Since the epidemic, we had significant decline that is ongoing. But for this 4-year period, 1/3 of the UK population. The chaffinch was lost. The chaffinch dipped and recovered. The Dunnock didn't decline. And if we go from those relative abundance estimates to actual best guesses of numbers of birds, we believe we've lost in excess of 1.5 million breeding greenfinches within a 4-year period. The outbreak is ongoing, and this is the largest disease outbreak in British wild birds on record. So it really shows that disease can cause a big problem of a common species in a short time frame. And we've worked with the ornithologists who understand about bird migration to show that we think it's migrating chaffinches. Half of our chaffinches leave in the spring to overwinter, particularly in Fennoscandia, and the disease was first seen in these orange spots in the summer of 2008. And these birds return, taking a slower path to overwinter in Britain. The disease was first seen in the Netherlands and Germany in 2009, 2010. It's now spread as far across as Slovenia. These are really widely distributed species. Wild birds move. It's an international collaboration that's required to understand the impact of these diseases. So just to recap, this is a parasite that was known to live in pigeons and doves and affect birds of prey. And our hypothesis as to why this emerged, or to how this emerged, is that there was a species jump of the parasite from a British pigeon and dove to a finch, but that once that's happened, the epidemic is finch to finch. And so we're currently doing molecular work to try and understand more about the different strains and move to a full genome of the parasite to inform these transmission routes. So why might this have occurred? The answer is we don't know for sure. The wood pigeon and other pigeon populations have been increasing because of the way that we manage agricultural crops. And as I've already shown, the greenfinch populations were increasing. And that might well be related to garden bird feeding. And we used to feed birds only in the winter months, but now we feed them year round. This disease happens during the breeding season or really picks up then. So all of those 3 factors might have increases the chances of the disease outbreak occurring. But it could also be that this is just a very aggressive new form of the parasite that's arisen. And that's why all of the focus is on the genetics of the parasite at the moment to understand more about that. So that's finch trike. And the last bit is avian pox. Again, really highly collaborative. So this is a completely different disease. This is a virus. This is spread through direct or indirect contact, often with abraded skin, or through vectors, mosquitoes in particular, mosquito bites. And it causes skin lesions in birds, often around the eyes, often around the beak. In some species, they often get better. But if they obscure vision or they interfere with locomotion, these birds may well fall victim to predation. And there's loads of literature on this. It's been reported across the world affecting many different species. But most of the focus is on the impacts of these island endemic species, these species with no natural immunity when the condition is introduced to those geographical regions. So, in Britain, since at least the 60s, there are reports of avian pox affecting these common British birds. And in these species, got a variety of pictures coming up here, often individual birds that are affected, and the lesions could be really mild. So this is on the leg of a wood pigeon, and you can imagine that this probably isn't going to cause a big problem to that bird. But here's a Dunnock. Now, this might be obscuring vision significantly. Here's a Dunnock with really quite severe skin lesions. a starling, again, with quite severe skin lesions. So in 2006, we had the index, which means the first case of a really severe form of avian pox affecting tit species. And I think you'll be convinced of that with these images. Now, over a 4-year period, around 3/4 of people that reported seeing these birds had multiple birds affected in their garden. So it's not individuals, it's outbreaks of disease. Most of them involve great tits. It's a staggering picture. But other birds, such as blue tits, are also susceptible. The feather loss is because the ultimate cause of death is a cat who's been able to get the bird and pull the feathers out. That's something that we see quite commonly. So a really, really striking condition, obviously adversely affecting the poor birds that have the disease. First step, again, traditional investigation. This is a slice of one of those lesions under the microscope, and these pink circles are the inclusion bodies, which are the virus production plants, very characteristic of this disease. We've taken the lesion, we've cultured the virus. Again, we've used electron microscopy to show that that's what's causing this problem. So, what's going on? Great tits are one of our most common birds. Estimate over 5 million of them in our back gardens. This is a really visible disease. We can be quite confident that this is something new. We would have heard about it if it had been happening beforehand. So the question is, where did it come from? And in order to answer this, we went to the sample archives that I mentioned earlier. Through the post-mortem examinations, when we've seen birds of these other species with the condition, we've saved the lesion, and we did molecular studies which showed that there were different sorts of the virus in different species. Apart from the dunnock, if we focus on that, we found in around 30 great tits an identical sequence, which was different to all of the other birds except that dunnock. So maybe this represents the virus jumping from British dunnocks to British great tits. Or maybe not, because the history to be aware of is this really aggressive form of pox in great tits has been seen in Scandinavia since the 50s, then there's a cluster in continental Europe, then we see it. So given this link in time and space, it seems very likely that there's a connection, and when we mapped out the incidence, we can see that they cluster in southeast England and spread out from that area. And that proximity to continental Europe makes an incursion of a new strain of the virus the likely explanation. So did that happen via bird migration, as the trike leaving Great Britain, we believe, did? Well, great tits are one of the best studied species. They're ringed and recovered. These red dots represent birds that were caught in Great Britain and recovered overseas, or vice versa. In comparison to 1.5 million ringed birds, there are a very small number of red dots. They don't move around much. So bird migration is not impossible, but it's unlikely. And instead we think either accidental human-assisted or wind-borne spread of the virus, of the vector, the mosquito, might be the likely route of spread. And again now, our attention is on the molecular aspects to learn about the virus and see why it causes this very severe form of the disease. So now on to the prediction part. You might have heard the University of Oxford has a very well-known long-term study population of great tits and blue tits in Wytham Woods. And this disease unfortunately reached there in 2009. And so they had a couple of years of really intensive study to look at the way that that disease spread and impacted the population. They nearly all have individual rings on their legs, so there's a lot of information to be gleaned. Loads of detail here, only one figure to focus on. Over that 2-year period for the great tit, 5% prevalence. That means 1 in 20 birds had evidence of the disease when they were caught over that period. So just remember 5%. These birds are repeatedly caught, so they can follow individuals and know that whilst there's a high fatality rate, they can recover. And they compared healthy and diseased birds in terms of their survival rate and saw a very highly significant decline, but most marked for youngsters And they were even able to look at the impacts on breeding success, which is extremely unusual for wildlife. They found that there was very little difference in the number of eggs laid when the parents were healthy versus when they were sick, but that there was a massive difference in the number of young that they reared to independence. And that might be because the adults get sick or that the youngsters get the disease too, or both. So given all of that information, again, a lot of national press, and the question, do we think we'll see a population level decline? And again, passing entirely over to the mathematicians at Oxford, they created a model to try to predict whether or not that would occur, taking into account all of that information that we'd collected. And this model predicted that a decline could happen, but it would only be if the prevalence exceeded 8%. We saw 5%. The model doesn't predict we'll see a decline. So that's the local picture, and then we take a step back and we use again that BBS data to look at how the populations change in A, the initial hotspot, in B, a larger area of southeast England. The box whisker plots, again, those populations changing over the 10 years before the disease outbreak. The coloured dots, what happens after the first few years of the disease outbreak. All of those coloured dots this time are within the box whisker plots. We've got no evidence for decline. So a real big welfare issue for the animals that are affected, but unlikely to be a conservation issue. An extremely well-distributed species, so again, a need for international collaboration to understand the disease impact. So, last slide. Those two examples, the details of those diseases don't matter, but I wanted to try and illustrate how vets and different scientists need to work with biologists to investigate the disease outbreaks, and to give you a flavour of, these are some really common, well-distributed, well-observed species. These diseases were known, but they've changed completely in terms of the species that are affected within only a 10-year period. The different routes of transmission, one's caused a really marked decline, which is ongoing. The other one is a welfare rather than a conservation issue. And so we need these projects in place for long term if we're to spot problems occurring and to determine if they're man-made in nature, if we should, or if indeed we can act in any way. So this is a bunch of funders and some collaborators, for which we're obviously very grateful, mostly grateful to the thousands of members of the public who take the time to get in touch with us and report sick and dead wildlife to gardenwildlifehealth.org. And I'd be very happy to answer any questions if you have them.